All right. Hi, everyone. My name is Zoe, and I'm one of the 201 IAs for anatomy. So I'm going to talk to you guys today about the neuromuscular physiology, neurophysiology, and nervous system histology. So I'll go ahead and get started. I'm going to share my screen with you guys once I find the page. Oh, that one. Okay, cool. So we're going to talk first about neuromuscular physiology. So the neuromuscular junction is a specialized structure that allows for communication to occur between a neuron and a cell/fiber that are interchangeable of skeletal muscle. So neuromuscular junction helps to communicate voluntary signals for body movement from the brain to skeletal muscles. So this is the whole idea behind how you're able to move, how you're able to pick up weights at gym, move your hands, stand up, turn your head, all of that movement, those voluntary movements are because of the neuromuscular junction. Because the movement involves motor or efferent signals, the neuromuscular junction is sometimes referred to as the motor end plate. So you can see that in this photo. Let me make sure you guys can see my arrow. In this photo right here, you can see the neuron synapsing onto the skeletal muscle fibers. Right there. Okay. Right there, you can see a little depiction of where the end of the axon, called axon terminal, is synapsing onto the muscle fiber. And we're going to explain this very, very detailed process of how movement happens. It's called excitation contraction coupling. So I actually have a whiteboard at my mom's house. Yeah, random, but I'm gonna go ahead and go over it on the whiteboard. As you can see, I have a very detailed flowchart that we're gonna go through. So I'll read it out loud. If it's hard to read, I'll make sure I read everything out loud. So it starts with an action potential. So this is excitation first. So an action potential arrives, and then the blank moves towards the presynaptic cell membrane, where it fuses with the cell membrane and releases blank into the blank. I'll give you guys a second if you want to think about what it might be. So when the action potential arrives, the synaptic vesicles there wasn't quite enough room right there. So the synaptic vesicles, here, I'm going to move it so it's easier to see maybe a little bit, move towards the presynaptic cell membrane, where it fuses with the cell membrane and releases acetylcholine, which I'm going to put ACH right there, stands for acetylcholine, into the synaptic cleft. Yes, this is also detailed in your lab manual, so if you want to follow along, feel free to do that. From there, if you follow the arrow, you'll notice it goes up here. So after acetylcholine goes into the synaptic cleft, acetylcholine will bind to the blank that are positioned on the postsynaptic membrane of the skeletal muscle. So what is that blank? If you said acetylcholine receptors, you are correct. So I'm going to put ACH receptors. Awesome. Okay. From there, the ACH receptors, again, acetylcholine receptors, open as ligand-gated channels and allow blank to enter the muscle cell. That blank is going to be sodium, which is Na+. Awesome. This begins the blank, blank, blank. That is going to be muscle action potential. So muscle action potential. Sorry, my handwriting is awful. But the muscle action potential is just the action potential within that skeletal muscle fiber. So if we move on from there, so now we have a muscle action potential, or MAP is its abbreviation. So we go all the way up here again. The muscle action potential travels down the plasma membrane to the blank. And that is going to be the T tubules or transverse tubules. I'm just going to put T tubules. The T tubules are just a continuation of the plasma membrane deep into the skeletal muscle fiber. So from there, this signals the blank blank of the blank blank to release its blank stores within the sarcomere of the fiber. That one was a little bit long. So this signals the terminal cisternae of the sarcoplasmic reticulum. So I'll start with that. So terminal cisternae of the sarcoplasmic reticulum. Again, if it's hard to read, try and follow along in your lab manual. Sarcoplasmic reticulum to release its calcium stores. So your sarcoplasmic reticulum holds a lot of calcium. Its calcium stores within the sarcomere of the fiber. So again, the muscle action potential travels down the plasma membrane to the T tubules. From there, that's going to signal the terminal cisternae of the sarcoplasmic reticulum to release its calcium stores within the sarcomere of the fiber. So that calcium now is going to bind to troponin C. Troponin C. That binding to troponin C, I'll put this one. That binding to troponin C, you can see we're going to go up here now, is going to cause blank to move out of the way and expose the myosin binding sites on blank. If you said tropomyosin, you are correct. Too short for this. Tropomyosin. So when calcium binds to troponin C, it causes tropomyosin to move out of the way and expose the myosin binding sites on actin. Awesome. I'm actually going to back it up a little bit and just go over this photo real quick. I drew out a sarcomere, so we can go over that so we can you can kind of see what we're talking about with troponin C, tropomyosin, and then actin, myosin, kind of see where they are. So this right here is a sarcomere. It's the most basic functional unit of a muscle cell. So it goes from Z disc to Z disc, which I draw it out right here. So again, a sarcomere goes from Z disc to Z disc. You have two really important proteins when it comes to muscle. You have actin and myosin. So the myosin is otherwise known as your thick filament, and then actin, otherwise known as your thin filament. So around the thin filament, you have this protein called tropomyosin, which you can see right there. I kind of drew like a going all the way around it. Because if you picture actin like just flat like this, you can see tropomyosin just swiveling all the way around it. And connected to that tropomyosin is troponin C, which I tried to draw on the like purple, but that's troponin C. So when calcium binds to that troponin C, it causes that tropomyosin to kind of swivel out of the way and expose these binding sites for the myosin on the actin filament. Okay. What else about this? You also have a few other areas. So you have your I band. Your I band is just showing the portion that is that has actin without the overlap of myosin. So all actin, no myosin. Your A band is just showing where myosin is, just one into the other. And it has the actin in it too. And then the H zone is just showing a portion of myosin without the overlap of actin. Okay. The M line is in the middle. You can think of it like the M line is where the myosin is attached to, right down the middle of the sarcomere. Yeah, that's about it. So this is a sarcomere. So this is now where we are. As you can see, let's just go over the last one again. So trypomyosin has now moved out of the way and exposed the myosin binding sites on the actin. So now the myosin heads can bind to the actin. Okay. So that was excitation. Now we're going to go into the whole contraction part of it. So blank on the blank head is hydrolyzed and cocks the myosin molecule at its hinge. So we have this molecule called ATP, is our body's currency. ATP. Cool. Yeah, you've probably heard it as our body's like energy currency. It is the energy within our body. ATP on the myosin head. So on this myosin protein, there's little heads that come off of it. There's a pretty good picture in your lab manual. I'll point it out when we're done with this. But so the ATP that is situated on that myosin head is hydrolyzed. So that means that ATP molecule is broken down into ADP and inorganic phosphate. So ATP is adenosine triphosphate. Tri meaning three, so it has 3 phosphate molecules. So that energy when it's hydrolyzed is the inorganic phosphate being cleaved off of that. So now it is ADP, or adenosine diphosphate, and an inorganic phosphate molecule. So that's where you're getting that energy from. So that causes it to cock the myosin molecule at its hinge. So now blank and blank can bind. So now actin and myosin can then bind. So let me write those down. Actin, myosin. Again, sorry for my awful handwriting. Actin, myosin can then bind. Blank dissociates from myosin and moves to its relaxed position, initiating a blank. So the first thing is inorganic phosphate is going to dissociate, or it's going to leave, which is just PI. Inorganic phosphate dissociates from myosin and moves to its relaxed position, initiating a power stroke. Awesome. Okay. Moving on. Okay. Blank is pulled towards the center of the sarcomere. So the actin molecule is going to be, or I should say protein, is pulled towards the center of the sarcomere. So up here, let's put actin. So it's kind of like when you're playing tug of war and you're like pulling on a rope, that's what's happening. So the myosin heads are pulling on the actin closer to the M line, or pulling the Z discs, because that's what the actin is connected to, closer to the M line, and therefore shortening that sarcomere. So actin is pulled towards the center of the sarcomere. Blank dissociates from myosin at the end of the power stroke. That is going to be ADP. ADP is going to leave at the end of that power stroke, and then blank binds to myosin head and breaks the actin-myosin attachment. So you need another ATP molecule to come and bind to the myosin head to break the actin-myosin attachment. So that's really interesting, actually, because when somebody passes away and their body is not resynthesizing any more ATP or energy, you're not producing any more energy, resynthesizing any more energy, that the actin-myosin attachment can't be broken, because you need more ATP to attach that myosin head to break that attachment. So actin and myosin are going to stay binded. So that results in rigor mortis. if you ever were wondering. That's why. Anyways, yeah, that's about it. If calcium levels stay high and there's still availability of ATP, then this process is going to keep on going. You're going to keep having that excitation contraction coupling. If the action potential stops and the calcium gets there's no more free flowing calcium within the sarcomere, then this is going to stop and there's no more contraction. Awesome. Cool. So I'll go back to sharing my screen. Okay, cool. So this is all basically what I was explaining. Yeah, right there you can see the myosin heads, you can see the myosin molecule. Let me make sure you can see the mouse again. And then the actin molecule. Okay. Yeah, so in all, the transfer of an action potential from a neuron to the skeletal muscle fiber, followed by the production of muscular force or tension, occurs almost instantaneously. So this is occurring very, 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 very fast. Step-by-step of series of events that make up this physiological process is known as excitation, contraction coupling, or EC coupling. Awesome. Okay. So now we're gonna move on to neurohistology and neurophysiology. So I'm going to share my screen again. There we go. Okay, nervous system histology and neurophysiology. So let's talk about nervous system histology first. So let's see, human nervous system is a complex structure as primary defining feature of what distinguishes humans from other species. So the nervous system has a lot of functions, including communication between various components of the body, coordination of body functions, response to our environment, either voluntary or involuntary, as well as higher cognitive functions such as learning, reasoning, our sense of self, or consciousness. So the basic functional unit of the nervous system is a nerve cell, or you can also call it a neuron. They're located within the CNS and the PNS, and we're going to talk about their basic histology. So I'm just going to go to the next page right here. So right here, you can see the neuron. So every neuron has three basic parts. That has a soma or cell body, has an axon, and it has a dendrite or dendrites. Right here in this photo, you can see that large portion is the cell body or soma. You can see a few of the dendrites, there's some dendrites, and then that portion right there is the axon. So the dendrites are what are receiving the signals. The cell body, again, soma, is kind of interpreting that signal, and then it's going to send it down the axon. It's going to send that signal down the axon. So the distal end of the axon is a synapse, where a neurotransmitter is released, and the next cell in the circuit responds to that neurotransmitter. So this is our body's sense of communication. Axons may be myelinated or unmyelinated. The myelin sheath, when present, insulates the nerve axon from surrounding tissue and increases the speed and efficiency of conduction. Awesome. So in this photo right here, you can just see the basic structure. So this entire nerve, this is how it's made up. So this is a nerve. Around it, you have the epineurium. Within it, you have separate fascicles. And around each fascicle is called the perineurium. And within that, you have a ton of different neurons. And around each one is the endoneurium. And then around each neuron, you can also see the myelin sheath and the Schwann cells that make the myelin sheath in the PNS. Awesome. Okay, I will go ahead and move on. So neurons can be grouped into three major categories. You have pseudo-unipolar neurons that have a single axon with dendrites at one end, and a synapse at the other end. So in this photo right here, you can see a pseudo-unipolar neuron. You can see the receptive endings, which are the dendrites. And you can see how it's one big axon. Yeah, cool. And then the bipolar neurons have one long dendritic process and one axon. So you can see on this side is the dendrite, and which it just has one for this kind, and this side is the axon. And then you also have multipolar neurons that have many dendritic processes and one axon. So right there is a good example of a multipolar neuron. It has a cell body, lots of dendrites, and then one big long axon. Cool. So yeah, this photo is just showing a presynaptic neuron and a postsynaptic neuron. There's a lot of little things within this photo too that you guys will need to know. So we have neurofibrils right there. We have the mitochondrion. You've probably heard it referred to as like the powerhouse of the cell. It resynthesizes ATP, very important. The nucleus, and then within that, the nucleolus. You also have right there, you can see the Golgi apparatus, that green structure right there. And then the nesal bodies, which are containing the RER and free ribosomes. Cool. The axon hillock right there is where the axon is starting. So the cell body, you can see it kind of like thinning down to one point, and then the axon coming off of it. So that is the axon hillock, and that's the axon. At the very end, you can see this little telodendria, little branches off of the axon, which are synapting or meeting up with the postsynaptic neuron right there. And the very, very end, you have the axon terminals. Cool. Next, the neuroglia or glial cells. So the glial cells make up a majority of cells in the nervous system. So glial cells have various roles outlined below, which support the structure and function of neurons. So if you go back to this photo, actually, you can see some neuroglial cells right there. And yeah, so the astrocytes are star-shaped cells with long projections that wrap around blood vessels of the brain and are an important component of the blood-brain barrier, which controls the substances from the blood entering the nervous tissue. Cool. So you can see right there, you can see astrocytes. Microglia are immune system cells that reside in the nervous system and act as phagocytes. So there's your microglia, immune system cells. Oligodendrocytes produce myelin in the central nervous system. So if we go down here, you can see oligodendrocytes. Again, they produce myelin around in the central nervous system. Oligodendrocytes produce myelin in the central nervous system. Whereas Schwann cells, right here, Schwann cells produce myelin in the peripheral nervous system in the PNS. Seem right? There. Nope, didn't mean to highlight all of it. Just the Schwann cell. Epidermal cells form cerebrospinal fluid and assist with the flow of CSF through the ventricles and fluid spaces. And you can see the epidermal cells right there. Awesome. Between the myelin sheath, there are these gaps called the nodes of Ranvier, and those are just gaps in the myelin sheath. And then between the nodes, so inter means between, so internode is just the space between the nodes. Cool. It does contain myelin sheath. Awesome. Okay, so now we're going to move on to neurophysiology. So this is going to be a lot of me reading and explaining the text. So nerve cells have two essential characteristics that allow them to carry out their functional role in the body, which is excitability and conductivity. So excitability means the nerve cell responds to a stimulus, so it gets excited. And then the stimuli can be either mechanical, it can be chemical, it can be electrical. And then conductivity is the ability of the cell to carry the excitation response along the length of the nerve cell axon. Awesome. So if it's able to, okay, I'll just because of these properties, when the nerve cell is stimulated, channels open the membrane allowing ions, which are charged particles, to flow across the membrane. This flow of ions causes a change in the net charge inside the cell, resulting in an electrochemical current that moves along the length of the axon. Cool. In this exercise, we will explore the electrical state of the nerve cell at rest, which is referred to as the resting membrane potential, and how that state gets disrupted or disturbed when a cell is stimulated, resulting in an electrochemical change called the action potential. Cool. The action potential travels down the axon as a wave of depolarization, which you can see in this image right there. You can see that arrow is the action potential. It's going down the axon as a wave of depolarization. And when it reaches the end of the axon, it triggers a series of events that result in the release of synaptic vesicles containing the neurotransmitter into the synaptic cleft. Cool. The neurotransmitter will then bind to the postsynaptic neuron or target cell and activate that cell. So you can see that in this photo right here. You can see that's the end of the axon terminal right here. And you can see those synaptic vesicles are going to move to that plasma membrane, fuse with that plasma membrane, release the acetylcholine into that synaptic cleft, and then that acetylcholine is going to bind to those receptors right there. I'll go over that again when I get here. But anyways, resting membrane potential. So the nerve cell membrane separates the intracellular fluid and the extracellular fluid. So this side is showing ECF is extracellular fluid, and this side is the intracellular fluid right here. So the voltage difference across the cell membrane arises from the asymmetric distribution of ions in those two compartments. So the extracellular compartment has a relatively high concentration of sodium, which is Na+, and low concentration of potassium, whereas the intracellular fluid has a high concentration of potassium, which is K+, and a lower concentration of sodium. This potential is maintained at rest by the sodium-potassium pump. And this results in the inside of the cell being relatively negative. So the inside of the cell is relatively negative at -70 MV, which stands for millivolts at rest. So then depolarization and action potential. So you can see in this photo, again, at rest, we're at -70 MVs at rest. Cool. When stimulated, the nerve alters its permeability to sodium. So sodium gates open in the membrane, and sodium rushes from an area of high extracellular concentration to an area of low concentration in the cytoplasm. Because sodium is a positive ion, this makes the inside of the nerve cell more positive, and the cell becomes less polarized than it is at rest or depolarized. So you can see right there, the millivolts are increasing, and once it hits that -55 MV, that is the threshold. So once it hits that threshold, there is going to be an action potential. This is an action potential. This you can see right there. So again, at rest, you're at -70 MVs or millivolts, and then once it starts depolarizing, when you get to -55 MV, that's the threshold. So if that threshold is reached, an action potential is going to happen. So that's the action potential. So that's depolarization. And then when the depolarization reaches a point that is irreversible, I just went over that, called threshold. Yeah, so once it reaches that threshold, it initiates action potential or nerve impulse. The wave of depolarization then moves down the length of the axon. The nerve cell then repolarizes as the membrane again shifts permeability, and potassium ions move out of the cell. Thus we see the action potential is a transient transient reversal of membrane polarity. So after depolarization, you have repolarization, so it's becoming more negative again. And then hyperpolarization, where it goes a little bit past that negative 70 MV, and then it goes back to that negative 70 MV. Awesome. Guess we'll move on to the synapse. So when the action potential reaches the end of the axon, it enters the axon terminal. I already talked about this. I just want to make sure I talk about it again, read the text. This triggers the opening of calcium channels. Oh, I forgot to talk about that. So the calcium channels right there are going to open, which in turn trigger the excitosis of neurotransmitters from the synaptic vesicles. The neurotransmitter will then bind to its receptors on the postsynaptic cell. Awesome. And that is all I have for you guys. Well, feel free to email any one of your eyes if you guys have any more questions, and don't forget to attend your lab this week. So yeah, have a great rest of your day.